Welcome to the SB Grid YouTube channel. Software tutorials by developers, lectures by structural biologists, unique content brought to you by SB Grid.
Hi, everybody. This is Jason Key at SB Grid. Thanks for joining today. This is the first one of these I've done in a long time. I don't think I personally have done a webinar since I don't know, January or February. So great to kick off, my end of the series here. Just a couple of announcements before we get started. In December, we've got a software title called Proteus from Thomas Simonson. So be sure to check that out. That's a relatively new one. And then in early 2023, we're going to kick it off with NMR from Chris Wadby, Titan. So that'll be cool. Then we've got FoldSeek coming up in February. We've got Model Angelo, this cool model building thing from Shosheris's group that I've had a lot of fun playing with. And then OpenFold. And then later in, toward the end of the spring, we've got another NMR title, Pokey from Woonhee Lee, which is sort of the successor to the Sparky line of NMR software tools, but it's much enhanced and improved. So be sure to check that one out as well. All right, great. With that, it's my pleasure to introduce Torsten Wagner, who's joining us from Dortmund. Torsten's a project group leader in computational structural biology at the Max Planck, and he is the developer of a few software titles now. I think you might know him from his particle picker Cryolo, which I think is our go-to particle picker here at Harvard Medical School. But he's not content to work in 2 dimensions, obviously. And so he's going to tell us today about TomoTwin CryoET, a picker for cryo-electron tomography. So go right ahead. Oh, before you start, just interrupt real quick. If you've got questions, go ahead and send them to me in the chat. You can send them to me and we'll save them up toward the end and there'll be time for questions. If there's something really pressing, I can interject, but we'll just save them all up for the end and we'll have time for discussion. All right, go right ahead.
All right, yeah, I'm also very excited to get the chance to talk again at SB Grid. Today I wanted to talk about TomoTwin. It's a generalized particle picking procedure for CryoET. And what we want to achieve with TomoTwin is to provide you a convenient way to perform particle picking and structural pattern mining in cryo-EM tomograms. As you might all know, those noisy tomograms are often super crowded and very heterogeneous, but they also contain plenty of information. And now if you try to train a common object localization procedure on those tomograms, you will quickly find out that the number of classes of objects of interest is huge, and it also might increase, also the number of classes might increase during the analysis of the tomogram. So to train common procedures might be very challenging and time consuming, especially because everything happening in 3D and you have to label the stuff. So for that reason, we want to go a different way. We want to find a certain representation of the tomogram that helps us to identify proteins quickly without additional training. So here we have our spinning spheres, and each point here represents one protein, one specific protein. And the left sphere here is the embedding of a tomogram with a mostly or completely untrained model. And the right one shows an embedding that we get when we apply our general picking model. And to generate this general picking model, we applied metric learning. So what we want to achieve with metric learning is the following. So assume you have your volume here with a certain number of objects. And now we extract at each and every position within this volume subvolumes of a certain size. So we have a running cube basically. And sometimes this cube might contain our object of interest and sometimes not. And after, so the next step is then basically we embed each of those subvolumes in a higher dimensional space and 32-dimensional space. And then we apply several rounds of metric learning. And we want to force a model to learn to put objects of similar classes close together, and objects of dissimilar classes, they should be put far away from each other. So once this is learned, we have two possibilities to find our object of interest. So we can either find a single reference of our protein of interest, and then we use, we can either use a certain thresholding or a nearest neighbor classification to find the remaining ones. Or we try to apply modern clustering techniques to detect those individual clusters. And TomoTwin supports both workflows. But before we discuss the individual workflows, I wanted to talk about how this is actually learned. So we apply the so-called triplet margin loss, which is almost classic, I would say, metric learning, but this is the loss that worked best in our hands so far. And during training, you provide triplets of subvolumes with a so-called anchor, a positive volume, and a negative volume. And the anchor volume and the positive volume, they share the same class, whereas the negative volume has a different class than the anchor and the positive. And to minimize the loss, it needs to pull the anchor and the positive class close together and push the negative away from the anchor. But it will not push it further away than a certain distance here from the positive, the so-called margin. And after training is finished, you hopefully end up with a nicely organized embedding space. But it turns out that this kind of training, you quickly run into local minima or convergence problems, because many triplets show a loss of 0 quickly. So to solve this, you need to apply a triplet mining strategy. So at every training epoch, you basically try to find those proteins that are not easy. So you have your anchor, your positive, and you only try to find negative ones that are in a certain region here, so-called semi-hard negatives. And those are used for training. And this helps the network to converge quicker. And the network itself, it's shared weights. And then you get those embeddings here. And after we have calculated the embeddings, you can calculate the distance between the anchor and the positive, and also the distance between the anchor and the negative. And having those values, you can calculate the loss and then run backpropagation to train the model. And now this part might be a little bit technical, but I would like to mention, because I found it really interesting, the network only trained in our hand when we use group norm. So instead of normaling across a batch dimension, the so-called batch norm, it divides it into channels. It divides the channels into groups, and then it computes per group the mean and the variance. And that surprisingly helped for all architectures we tried. I found this very interesting and surprising. But anyway, now let's talk how we actually got the training data. Because as I said, generating real-world training data is not really feasible here, as it was, for example, for Bryolo. We hardly have any labeled data and that we could easily use for training. So we needed some kind of different way to train it. And what we did instead is we simulated tomograms. We created 12 sets of simulated tomograms with 10 different proteins per set and up to 150 particles per protein. So, and when we selected the proteins that we want to use for training, we made sure that they are all pairwise dissimilar. And we did that by calculating the density maps for each individual protein, and then we scaled them to 10 angstroms. And then we calculated all possible pairs, aligned them to each other, and calculated the cross correlation. And then we only took those PDBs that have a minimum similarity below 0.6 to every other protein in the set. And then to measure the generalization, we created a separate tomogram that contains proteins that are not in the training set, but because what we actually want to know is how does it apply to new proteins? We don't want to pick those proteins that we trained on. All right, having those proteins, we start simulating the tomogram. And now let's look at how the model that we got actually performs. And TomoTwin provides two workflows to do the picking, a reference-based workflow and a clustering workflow. So the reference-based workflow works like this. You have your tomogram, you have your generalized model, and you pick one single reference for your protein. You can also pick two references per protein. That doesn't matter, but you need one reference. Then you embed the tomogram and the reference, and you get tomogram embeddings, and you get your reference embeddings. Those two, so the embeddings go both into TomoTwin map, which basically calculates a probability map where TomoTwin thinks your protein is located. And TomoTwin locate is then identifying the individual hotspots where your proteins could be. And then you can use our Napari plugin to look how the pics actually look like and filter them as you want. The second workflow is a clustering workflow. Here you see the reference disappeared. You still have your tomogram and the generalized model as input. You calculate your tomogram embeddings, but then you project those high-dimensional, 32-dimensional embeddings onto a lower dimensional space. And for those who are interested, this is a classic UMAP. So you have your two-dimensional map here, and then you circle clusters that you think that might be interesting. So you circle those clusters, and then for each of those clusters, you calculate one average, basically, and that is your target embeddings. So for 3 clusters, you get 3 target embeddings. Then again, you calculate the probability map using TomoTwin map, and with TomoTwin locate, you can then identify the positions and check them using Napari. All right. I think now we look at some results for the reference-based workflow if we apply it on our validation data. So first of all, we get a pretty high median F1 score of 0.92, but you also see some outliers here. But most of them are located at, let's say, high F1 scores above 0.7. This you can only also see at the general distribution here. Here's the F1 scores, and these are the number of proteins, and you can see many of them are in this regime where I would say these are high F1 scores. But still, we have those outliers. And then we look a little bit closer to those outliers, and it seems not to be dependent on size, it's rather dependent on contrast. So here, for example, we have proteins of similar size. One is not really well picked, and one is pretty well picked. And the difference is that this one you can identify, and this you hardly see in the tomogram. There's basically no contrast. And the same applies for other proteins. So this is still something we look into. It might be interesting to see what happens if we use denoise tomograms and so on, but this simply needs further investigations. We also compared it to template matching. So we took template matching, and then for each protein, so basically I used a couple of thresholds, and we optimized several parameters using a grid search, and then I reported the best F1 score I could find was template matching. And for TomoTwin, we just applied our normal reference-based workflow and optimized the threshold for the proteins. And you see that if you look at the distribution, the proteins are highly accumulating in this area where you have high F1 scores, whereas for template matching, it's much broader. But these were on our validation data. TomoTwin was trained on this. We are not, in principle, we are not interested in that because we want to know how it performs on the generalization tomogram. And here, for the tomogram we simulated, we got a median F1 score of 0.85. And F1 score for the biggest protein is 0.99, so almost perfect. And the smallest protein, 0.88, which is still high. We furthermore analyzed how the performance increases with a training data set. So we started with 20 proteins, and you see the spread is quite huge. But as we add more and more proteins, it gets better and better. And between 100 and 120 proteins, you hardly see a difference. So we say, okay, that is enough proteins. And that was basically the final training set size. But still, while this is at least not in the training data, it's still not super interesting because it's not real world data. So, or not real tomograms. So the next thing we did was we created a mixture of 3 particle populations. Those mixtures contained TCDA1, aquaferritin, and RHSA, which is a type 6 secretion effector protein. So even though the network was not trained on any real world data and also not on TCDA1 and HSA, it did a really good job and picked them with high accuracy. But still, while this is already very promising, it's still kind of artificial data because it's more or less a single particle grid where we collected tomograms from. So we actually wanted then to know, okay, how does it perform on cellular tomograms? And here's one result that we produced. We applied it to plant cells to identify Rubisco. And what we choose that sample here as it was also used by the deep learning tool DeepFinder, which produced excellent picking after training it with thousands of samples. But in contrast here, we only picked a single reference. Then we run our reference-based workflow. We got 30K picks. We processed it, and we end up with basically the same resolution as they had, but without any training. Now let's look at how you can play with your data in the Napartic UI. So here I created this small video. Let's start it. What? I'm not sure why this is not starting. We were testing it just before. That is a pity. Why is this not starting anymore? Okay, I hope that the next video will work. So what we did, we run the picking here, and then we optimized the threshold, and we got nice picks, we process them, and we end up in the ribosome clusters. And I think this already indicates that picking was quite accurate. So the next thing I would like to talk about is the clustering workflow. So we took our generalization tomogram, we calculate the 2D representation, and we found our clusters. So we selected those clusters, and each of those clusters corresponds to one of the proteins in the simulated tomogram. There are more clusters, but it didn't give anything meaningful. So that already works quite good. And if we compare it with template matching, you especially see here for the small proteins, it outperforms template matching by quite some margin. We also applied it to the ribosome tomogram. We selected this single cluster here. We did the picking, and we got the ribosomes. So here we apply tomogram to a bit more complex tomogram. We calculated the 2D embedding. We selected 2 clusters. You already noticed there are more clusters, but this didn't give us anything interesting. We processed both clusters, and one cluster was representing the ribosomes, and the other one is probably RNA polymerase. We don't know yet, but at this resolution it's hard to tell, but it looks like it. So now let's look at this video here. And this is also not working. Maybe we restart this presentation, maybe because it was working before. Jason, what do you think?
Sure. Yeah, if you think.
Now it works. Perfect.
All right, great.
So
you just have to talk about restarting it. You have to threaten to restart it.
Yeah.
And then it starts working.
Here you see the tomogram. Perfect. So now what I do next is I'm loading the results that TomoTwin produced. So we have 2 references here. I select one of them, and now I adapt the box size to a more reasonable value, and then you can start filtering it. So I adjust the confidence threshold, and you can be more strict or less strict. And I find one specific value, and basically that's it. Wait a second, I'm not that fast. Okay, and here that is the second value that you can optimize. It's basically the peak size. It's sometimes helpful to get to remove spurious peaks, but here it basically is not necessary to filter. And these are basically the particles that went into the refinement for their RNA polymerases. All right, the last couple of slides, I would like to talk about a tutorial, basically how you actually apply it to your tomogram. You start, and this is actually an optional step. You start by filtering your tomogram down to 10 angstrom. Why? Because TomoTwin was trained on data with 10 angstrom. Anyway, we also run it on tomograms with a pixel size with 15 angstroms or a bit smaller than 10 angstroms. That always work fine. So it's not always necessary. But for example, if your particle does not fit into the box of 37 times 37 times 37, which is our input volume, then you might have to use a bigger box, a bigger pixel size, like 15 angstrom or something like that. All right, this is step 1. Then you start NAPARI. You select one of your references here. I select the toxin, and you extract this reference in the next step. So NAPARI, you save the coordinates of your reference, and you extract the coordinates. Then you have to embed your tomogram, and you have to embed your reference. 2 commands, and it's quite straightforward. You get your embedding for the tomogram, and you get the embeddings for the references. So now map is now calculating the probability map, and I hope this is working now. Yes, it does. You see those peaks that we have to now locate in the next step. So here you run the locate command, you provide the probability map, and you end up with your located particles. And how this whole process looks like is shown in the next video. And it's working. So you select your reference, you run the process, you load the tomotrin results, and here are your picks. And if you check the results, these are all toxins, which is great. Current limitations, we only trained it on 10 angstrom. We plan to diversify this. Proteins needs to fit in the 37 times 37 times 37 box. If that is not the case, you can help yourself by choosing a different pixel size. The model is not trained on denoise tomograms yet. That was we will explore how this will help the model. And membrane bound proteins can't be selectively picked, while if it's visible, it probably gets picked, but we can't say only pick proteins that are attached to a membrane. That is not possible yet. We can't pick filaments, and right now you can't use PDBs for picking, but all this we want to explore. So right now we have our official release. We are working on a streamlined clustering workflow and tutorial. I would like to provide some developer information in case someone wants to contribute. Then we would like to work on the PDB mapping, which simply means that I can use a PDB for picking, which would be cool, and some unsupervised clustering for more automated processing. And in the far future, we would like to work on membrane models and filament picking. And while we do this, we will constantly do model improvements. That means we could develop different architectures that might work better as the current one. We will add more simulated data and maybe also real-world training data. On the GitHub repository, you can find all the source code and look into it. You find any information you need. There is also a link to the TEM simulator scripts. There are a couple of scripts to streamline the generation of our simulated tomograms. We use the tool TEM simulator here, but we simulated that many tomograms that we had to streamline it, and that is basically a result of it. All data that we use to train tomotrin is public available, and if you want to play with it, there is the documentation, and there's also tutorial. And yesterday, I also put an example data set online that you can directly use and see if everything is working for you. For more details, please check out our preprint. And the last thing I have to mention is a big thank you, for example, for Stefan. He's the director here and provides us a lot of support. Gavin and Marcus, who are my co-developers. Gavin and me were mostly working on the machine learning part, and Marcus developed basically the Nupari box manager. Of course, thanks the whole lab for testing this stuff, and I'm looking forward for questions.
Well, that's great. Thank you very much. That's fantastic. Questions, we had a few in the chat. We can also take questions. You can raise your hand with the reactions button, and it should bump you up to the top of the participants list, and I can call on you. Let me unmute. I can allow people to selectively unmute themselves if they want to ask a question. All right, we are now unmutable. So Any questions? We had a few. Daniel Mann had one in the chat. Daniel, if you're online and you've got a mic, you can unmute and ask yourself if you prefer, or I can read from the chat. Go ahead.
Can you hear me?
Yes.
Awesome. Hi, Torsten. Thanks for the great talk. So I was wondering about the missing wedge and how that relates to particle picking accuracy. So my first question is, did your simulated data contain a missing wedge?
Yeah, we simulated from -60 basically. We have a missing wedge that is typical for the tomograms. So we basically set it up like the same way we did it in-house.
Awesome. And can you see an effect on the picking accuracy if you, I don't know, take the missing wedge away? Like, does that help?
Actually, that is we did not look into this. Look into that question. It might be because it should be easier to generalize. I could imagine if there would be no missing wedge, it could work better. And I mean, with tools like ISONET, it might be possible to generate data that is similar to non-missing wedge tomograms, but we haven't tried it yet. But it's a nice suggestion.
Matt Larson, go ahead.
Hey, Thorsten, so you mentioned that we should downscale to 10 angstrom for the processing. Can you be lower resolution? So if you had bin data, can you be at lower than 10 angstrom binning, and that will also work? And then if you want to take the coordinates from the picking and bring it back to something that's at a higher resolution, is that possible to convert through some script?
Yeah, so we provide, we have this Tomotwin tools that comes with Tomotwin, and this also provides a rescale command to do that. We also picked tomograms that with a pixel size lower than 10 angstrom, so that is working. But still, our experience in that regard is quite limited. So we will see how it performs in practice if we applied it to more and more tomograms.
Great.
Thank you.
Thank you. Joe Dobbs had a question in the chat. If you want to ask directly, feel free.
Yeah, please. I just thought maybe the video is not working and I could show you again, but I'm not sure.
Sure. I can read Joe's if he is unmiked. He was asking, how does Tomotwin resolve complexes where one view looks very different from the other? Top versus side views, do they still cluster together? Yeah,
I mean, that is a good example for this was the last video I showed you with the TC toxin, which has a top view that looks pretty much different. And I mean, we don't train on the 2D views of them. We don't train on the individual slices. We train on subvolumes. And while we are training, each protein example, basically in the simulated tomogram, has a random orientation. So the network should have learned that it does not depend on the individual orientation of the protein. Of course, we can't guarantee this, but for example, the TC toxin is a pretty good example that it's not dependent on the orientation of the protein.
Great. Any other questions? Feel free to raise your hand or jump in the chat. I have a sort of elementary question that I just was sort of wondering as I went through the documentation. Where does the 37 by 37 by 37 come from?
That was in the very beginning where we started to use that because that was in our training set at that point. It was the biggest protein we had, and with 37 times 37 times 37, it worked nicely. Later on, it turned out to be a bad decision because it made some design of the software hard. So probably in a later version, we might increase it to a not prime number because this was really not such a clever idea. But now it is as it is. We will probably change that with an upcoming version.
Yeah, I was wondering if there's some sort of mathematical benefit to having a prime number, or
no, it was just a stupid idea.
Yeah, it's, as somebody who's, always thinking, with FFTs sort of in the back of my thinking, I'm thinking, okay, well, maybe that's probably not the best way to go, but I don't know. Yeah.
It's just stupid.
Yeah.
That's all.
It's interesting. Great. Any other questions? Anyone? All right. Well, I will just let everybody know that Tomo Twin is in SP Grid. It's brand new, so we haven't rolled it out to everybody, but it's ready to go. It includes the Napari viewer, and there's a brand new tutorial to work through. The tutorial set up with a convenient RunMe script, and it's set up to run on 2 GPUs, but it's trivial to see if you want to modify and run on a single, and it looks pretty easy to power through. So I love to have good tutorials. It's always a good way to get people started, especially with with new sophisticated software tools. So Torsten, thank you very much.
Thank you.
Anyone who's
And thanks for your attention.
Sure. Great. And thank you everybody for joining, and see you next month.